So, dietary lipids are those lipids which we eat on daily basis, which is part of our diet. Generally speaking, adults consume almost 70 to 80 grams of lipids daily. The lipids which we consume or which are part of our diet are predominantly, a major part of them is composed of triacylglycerols or triglycerides. Almost 90% or over 90% are triglycerides. The triglycerides consist of fatty acid and a glycerol molecule. Then there are other lipids also which are part of our diet, though to much less extent, but still they are part of our diet. For instance, cholesterol and phospholipids. When we take food through our, by our eating process through our mouth, and of course the digestion process starts straight away in the mouth. As far as lipids are concerned, the enzymes which are involved in the digestion of lipids are known as lipases. In the mouth, there are lipases, which are specific for the mouth, or specifically known as mouth lipases or lingual lipases, because they are released from the back of the tongue. And then there are gastric lipases also, because when the food goes towards the stomach, it is acted upon by lipases. But keep in mind that these lipases don't contribute much towards the digestion of the lipids. Most of the digestion of the lipids takes place in the small intestine. Also, infants, because infants contains many of the lipids components which are of intermediate size or short length fatty acids. So in infants, the digestion by the gastric lipases is very important. It's also very important in those people in which the pancreatic lipases are not really, they are impaired, they are not working well. So that's in them also the importance of these lipases, the gastric lipases, increases. Once the lipids come into the intestine, the duodenum, the small intestine, then a very important process takes place that is known as the emulsification. Emulsification is the process in which the larger particles of lipids are under the action of the enzymes, specifically the pancreatic enzymes. They are divided into smaller parts so that the surface area for the action of the lipases is increased. Then there are also, in addition to the lipases, also the bile salt also contributes towards this digestion in the intestine. The bile is a product of the metabolism of the cholesterol. If you can see here, so this is a sort of, we can say, a bile molecule, and it is a conjugated molecule because it is made up by the conjugation of a cholesterol with glycine. So in this, this is a cholic acid, this part, and then this is the glycine. So in this way, the cholesterol is not only important, doesn't have its own importance in signaling and in the structural making of the cell. It's also is very important because it's involved in the formation of bile, which we will study when we will be talking about the metabolism of cholesterol. The bile is stored, of course, in the gallbladder. It is made up in the liver, but it is stored in the gallbladder. I will like with you to share a very important link here, which is an animation showing how the emulsification process takes place in our body, specifically as far as the digestion of the lipids is concerned. So this is this video link will be available to you through these slides and through the text which will be provided to you also. So this is a very nice animation video which I found for you guys. Please watch it carefully. And I'm sure, because I always say to my students that a figure covers 1,000 words. An animation, a video animation, is a substitute for millions of words, because then you can really understand much well. So I will like that whatever animations, whatever videos, their links I share with you, you go through those videos and focus on them. Then the pancreatic enzymes, as I have told you, the major enzymes which are involved in the digestion of lipids are the pancreatic enzymes. They act upon the triacylglycerols, they act upon the cholesterol, they act upon the phospholipids, and then divide them into their constituent compounds. So, for instance, the triacylglycerols are concerned, the pancreatic lipase, this one, acts upon the triacylglycerol, and then we get, as a result of this action, 2-monoacylglycerol. This 2-monoacylglycerol is a compound in which have lost its 2 fatty acids because of the action of the pancreatic lipase. These 2 fatty acids are gone, so a fatty acid at the carbon atom number 1 and at the 3, they are lost, and you end up with glycerol derivative molecule, which is triacylglycerol derivative molecule, which is composed of the glycerol moiety and 1 fatty acid. Other 2 fatty acids are released. Another molecule which I should mention here is the colipase. It actually contributes in its function by facilitating the action of the pancreatic lipases. A drug known as orlistat, actually it is used as an anti-obesity drug to reduce the fats in the body. It acts by inhibiting the actions of gastric and pancreatic lipases. Then there comes the cholesterol ester degradation. Again, I have told you there are 3 important lipids: the triacylglycerides, the cholesterol, and the phospholipids, which are acted upon by the pancreatic enzymes. So what happened that cholesterol ester is acted upon by cholesterol esterase, and as a result of this, we get a simple core cholesterol molecule which contains its core steroid, what we say core sterol moiety, and in this way you end up with a cholesterol moiety and a free fatty acid. Of course, because I have told you that bile salt and the pancreatic enzymes both act upon and synergize the action of each other when it comes to the digestion of the lipids. Then finally there comes the phospholipid degradation, another very important lipid. You know, your cell membrane is made up of phospholipids, very much made up of phospholipids. So the lipase which involves the phospholipase, it comes again from the pancreatic juice. It shows the importance of pancreatic juice, that so many enzymes it contains for the digestion of the lipids. So what happened, if you can see here, this is a phosphatidylcholine. It consists of the glycerol moiety and the carboxylic acid and the fatty acid moiety. So what happened that as a result of the action of this phospholipase, 2 of the fatty acids are released, and you end up with the glycerophosphatidylcholine molecule, which contains only 1 fatty acid. So in this way, as a result of the action of the enzymes, what will happen that you end up with lipid molecules which are smaller in size, so that they can move towards their further destinations for further catalytic processes. As far as the hormonal control is concerned, 2 important hormones, CCK and secretin, they contribute positively towards the digestion of the lipids. And as far as secretin is concerned, there is another very important aspect of secretin is that, when your food is coming from the stomach into the intestine, what will happen that it may have acidic content. The medium can be acidic. So to make it neutralize, this secretin releases bicarbonates in the solution. So as a result of which, the solution or overall pH of the medium become more basic. So as a result of this, the acidic environment stimulates secretin, which then in turn make the environment more basic, which is important as far as the digestion is concerned.